Okay. Hey guys, it's Ilya. So I figured I'll give you a little bit of, I'll give you the presentation that will explain how one of the therapies we're trying to push forward works. Before we begin, just so we know, these are not some fancy glasses. These are actually my vision glasses, and so just in case you had that thought, now you know why I have it. So I'll explain to you a little bit of what it is that we're doing right now. But for you to, I guess, understand it better, I'm gonna dive just slightly into the physiology in the cell physiology to explain how it works and how the problem persists. And then I'll also give you the solution that we're trying right now, and hopefully we're gonna push into clinical trials very soon. So in the middle, you see something called a cell, right? This is a cell. Within the cell, you have a nucleus. In the nucleus, you have a DNA. And usually you will have two alleles or two strands. And in our case, what we have is we have an error in one of the alleles, and this is called heterozygous mutation. So for demonstration purposes, you see that this is in red, and this is the error that our children have. And this other allele is actually pretty good, has no problems at all whatsoever. So what happens in general is that you have all these cells or neuron cells in your brain, and they all communicate to one another. And they use something called receptors. So in this picture, you will have receptors here. And then I'll kind of go into a breakdown and explain how receptors actually work. Again, this is very high level. I'm trying to abstract a lot of things here to simplify it, hoping that many of you will understand what's going on. So again, you have a nucleus. Within the nucleus, you have the DNA code. So whenever a request comes in into the cell and says, hey, we need to produce a GNAO1 protein, what happens then is the DNA is being read into a smaller subset, a smaller code called RNA. And usually, so DNA would be something this large, and RNA is a smaller, shorter version of what it is we need to create. And what you will see here is that RNA, it's broken into two stages. You have the one in red, which is something we call toxic. This is a toxic DNA or toxic RNA. And then the one in the green is called wild type. Let me just note that here. And so the wild type is a good one, and the toxic is the one that causes all the problems. And so just so you understand what happens. So this creates that, and this creates this. So the call comes in, the DNA is read, a shorter version of the DNA is then translated into something called RNA, and eventually you will have protein, GNAO1 and G alpha O proteins. Because of the genetic error, you will have a protein that is malformed. And so instead of it looking like this, which is the way we would expect it to look, it looks like that. It's toxic. And I'll explain what it means. So we're going to go one step further into receptors. These are the receptors I have. Now, it's kind of hard to understand what it looks like, so I'm going to give you an analogy of the receptors here. So just think about receptors as the game that kids play with pegs, where they have to take different shapes and they have to smoothly go in. And so over here you have two sides of a receptor. In case of our children, because they have a genetic mutation, a toxic G alpha O or GNAO1 is being created. And that toxic G alpha O, you can see right here, has a bulge, right? And that's the problem. Because of the letter or specific error, it looks different than what's expected. And the toxic protein goes into the receptor and it clogs it. And it prevents the good wild type proteins going through the receptor. And this attributes to the issues many of our children have, where they don't develop, or they have low muscle tone, or what can happen in some cases is that, this toxic protein will eventually expire, and then you'll have a surge of wild type and more toxic proteins go through these receptors and overload them, and that could cause seizures or movement disorders. And so, one of the projects we're trying on right now is we're trying to use something called antisense oligonucleotides. I just wrote antisense oligos here for short. Now I'm going to refer to them as ASOs because it's just much easier. And so ASOs have been out for quite some time. I think they were discovered back in 1970s. Though in the past 5, or let's say maybe past 10 years, they actually became more mainstream, and they're being used right now in many targeted genetic diseases. Huntington is one of them. Others, quite a few. I can post links if you're interested. So the way antisense oligos work is that they contain something called guide RNA. Guide RNA is literally a sequence of letters that you see here that contain your RNA code. So again, I'm going to simplify this one step further. In case of my son, he has, and this is a DNA code, but I'm just going to pretend that it's an RNA code. He has 607G2A, and that means a position of 607, which is in this case here, the letter that was expected to be G became an A, and that's what causes that toxicity. That's what causes creation of this malformed protein. So the guide RNA in antisense oligonucleotides contains this sequence. Now we create a very small synthetic molecule and we inject it into the spine, which eventually gets into the brain. And what this drug will do is that it's going to go and it's going to bind into a cell right here. Right? This is the same thing. I don't have enough space here. And so this drug, a tiny drug, will bind to neuronal cells everywhere. And the next thing it does, which is interesting, it will read the RNA sequence. And it's going to look for this toxic sequence. And the moment that toxic sequence is identified, it's going to invalidate this protein. It's not going to create this toxic protein. And so what you're going to end up with is literally only good wild type proteins here. And that should remove the toxicity and allow for our children to hopefully begin developing and function, address the seizures and movements or any other issues. At least this is the working theory right now, and that's what we're trying to push forward. We do have a team of about 10 people right now that are working on this, and we're making progress. But the reason I'm showing it to you is that I hope that this presentation will explain what it is that we're trying to do, and also hopefully you will now be more versed in understanding a little bit of genetic therapies. So thank you so much. If you have any questions, feel free to ask. Have a good day.